Hello all, this is Dr. Jayesh Thalani and I am going to introduce you Bio-Analytical department. So here this is my first video, introduction to Bio-Analytical method validation. Generally, most of the people are confused between the analytical method validation and bioanalytical method validation. So here I am first clearing you the analytical method validation is deals with what aqueous solution, and bioanalytical method validation is deals with blood, urine, plasma, etc. Means related to blood or anything. So first of all, the introduction to bioanalytical method validation. There are why it is required. So first I am introducing you to bioanalytical method. Bioanalytical method is generally used to determine concentration of drug or metabolites or both in biological metrics as plasma, blood or urine. You can say that's why it's called bioanalytical method validation. So why it is required? There are two parameters are main. Because of that, bioanalytical method validation is required. First is the BA and second is the BE. Bioanalytical information is used to prove the BA and BE. What is BA? Amount of substance that becomes available to organism body for bioactivity when introduced through injection, oral or any skin contact. So that is called bioavailability. What is bioequivalence? Means you are proving that these two compounds are biologically equivalent. Actually, this is the pharmacokinetic parameter, and this is all in vivo. We can say that this is in vivo parameter. Okay. Next is, so most of the people don't know the why BAB analysis or study is required. They are just knowing the theoretical aspects. So here I am explaining you the main aspects or main practical aspects of bioanalytical method validation. Here I am taking one example. Suppose one company has made the novel molecule that is called ethinyl estradiol and they have patented. So for some years, only the X company make and X company makes that ethinyl estradiol molecule and they can market that molecule. Now that molecule is patented, so the no one can synthesize. So why company wants to synthesize that molecule? So they will wait up to the patent expire. After patent expire, they will synthesize the molecule, and after expired of patent, they will synthesize the molecule. They will prepare a formulation according to BA. So no company will provide the information regarding formulation. So they, if you want to market that ethinyl estradiol drug, so you have to make your own formulation according to BA and BE. They will submit their formulation samples to bioanalytical department or any contract research organization. Contract research organization, they will develop the method and prove that BAB studies and submit all documents to regulatory. After approval from regulatory, the Y company market that ethinyl estradiol drugs. So this is the all funda behind the bioanalytical method validation. You can see here. So how Y company prove that the X company's ethinyl estradiol and my ethinyl estradiol are same. So first ethinyl estradiol taken as a reference, and second one Y company's sample is taken as a test sample. How it performed? So how we can say that the BAB parameters are same for reference and test sample. So there are number of healthy human are invited for this study, and first sample will be collected as a pre-dose sample. So why it is pre-dose sample? Because we have not given any dose, that's why it's called pre-dose. And there is no concentration or zero concentration of drug available in that sample. So we can prove that the patient is healthy human, and there is no drug taken by him or her. Next is the reference drug given to the human. As per the decided dose, as per the protocol, the reference drug is given, means X company's drug is given. After that, as per the protocol, as per the bioavailability, like 24 hours or 48 hours, 76 hours, as per the bioavailability, and as per the protocol, during after the some time, like 30 minutes, 60 minutes, 120 minutes, this is called time gap. According to protocol, according to time gap, the blood samples are collected. So you can see the we have given the dose of drug to the healthy human, and we are collecting the samples. So you can imagine the when drug is dissolved in the body, and drug concentration will increase in the blood samples. So after the collection of all samples according to protocol, what happened? The all the samples stored in the deep fridge according to drug stability. Suppose it may -20, it may -70. Next, after some days, means you can say the 15 days, 20 days, whatever the time decided, healthy human called invited again, and test drug sample will be given to same human. Now you can say that same protocol again followed. 5 ml blood collected as a pre-dose time point. Again, according to protocol, point 5, 30 minutes, 60 minutes, 120 minutes, whatever the time get given in the protocol, the sample is collected. All samples are stored in deep fridge. So you can say that the reference sample and test sample you have now. Now what is our target? To analyze that sample concentration. For analysis of concentration, we have to retrieve the samples from deep freezer. You can say that the samples are freezed, so we have to give the time for thawing. Approx 45 to 50 minutes is times required to thaw. Now we have a concentration in blood, or we can say in the plasma, according to protocol. If or in protocol written, drug is in the plasma, if blood is drug is in the blood, whatever we have stored in the deep freezer, we can retrieve. Now, on our next target is we have to extract our drug from that sample. So we required extraction method. So there are three methods which are used in the bioanalytical department. First one is the solid phase extraction method, liquid extraction method, and protein precipitation extraction method. What we are doing by extraction, and we are making the sample compatible to the instrument analysis, because instrument will not directly analyze samples from the plasma. I am talking about the same instrument in terms of LC-MS, which is nowadays huge used in the bioanalytical department. So if we want to analyze sample in the LC-MS, so we have to take a drug which is soluble, which is available actually in the plasma or blood sample. So we have to extract. So first method is the solid phase extraction method. So solid phase extraction method is why there are many advantage of that method. Solid phase extraction gives us good recovery, and very less solvents are used, and by this method the many times instrument after more time instrument cleaning is required compared to other method. Liquid extraction method, as you all know, it is depends on the partition coefficient. Compared to solid phase extraction method, the recovery is minor less in liquid extraction method. Protein precipitation extraction method. In this method we are just precipitating the protein. So protein binding, if the protein binding is more than 90%, then I can suggest this method. So after extraction, we are going to inject the sample into the instrument. So here you can see these two part require method development and validation. So this is on the basis of result, we can say that we can prove that BAB studies. Steps in method development. The first step is literature search for the drugs. This is the very basic requirement. Development of analytical technique. So the development of analytical techniques in terms of extraction method, in terms of you can say that chromatography technique, reference standard. Reference standard is required for the preparation of linearity. You can say that the linearity, we want to analyze the unknown samples, so that's why reference standard is required. And now, why internal standard is required? So internal standard is generally same structure as a like reference standard, but some some deuterated structures are used. Nowadays, reference standard is mostly deuterated and it's called as the internal standard. So next is the sample pretreatment, the extraction and recovery of analyte. It's called the sample pretreatment. Next is the sample storage. Sample storage conditions is defined in the method development. You can define sample storage conditions like -20, -70 in method development. Literature, what you can do in the literature search. So first one is the what are the formulations available, what is dose. According to you can define the linearity concentration, Cmax, Tmax, T half, how much protein binding is there, the structures, molecular formula, pH and solubility of drug. Depends on solubility, you can identify the mobile phase. Identification of analytical techniques and optimization. So basically we required good chromatography for analysis. For analysis we have to identify the conditions which are compatible for good analysis. So which parameters, like column, there are many options are available in column. Second one is the mobile phase. Mobile phase, you can see the like analytical method development validation, you can identify the mobile phase. uh, on the basis of trial and error. pH of mobile phase, you can say the buffer of mobile phase, temperature of instrument, temperature of lab, temperature according for the stability of drug, flow rate of instrument, method of extraction. I already told the liquid extraction, solid phase, and protein precipitation. These are all the steps of method development. What is validation? Validation is nothing, and many people are confused in the validation word. So, if you are mugging up the definition, so you will be the confused. Just I want to tell you in the very simple word, that is, validation is the we want to develop some method. Like if you have developed the clinic method, so how you can say that the my cleaning method is very appropriate, so that requires some confirmation, so that confirmation called validation. According to validation, you have to define some experiments, and on the basis of that experiment results, you can say that my method is accurate and my method is precise. So here you can develop the analytical procedure, uh, which is when analytical procedures are developed, and there is a sets of experiments given in the guideline by regulatory, and you can say that after the experiments you can say that my method is accurate and precise. Now, what is the requirement of validation? By definition, you already understand the accuracy and precision is requirement by the regulatory, and as we are dealing with the human being, uh, so the accuracy and precision is required, because a simple analysis is not important, but results, reproducing result and accurate results are very important. So that's why validation is required. Bioanalytical method validation, we can say in three parts. When we are doing the first time, at that time full validation is required. When we are changing the minor in the range or whatever the some experiments we required, at that time we can perform the partial validation. Like partial validation, we can perform the cross validation also. There are sets of conditions are defined in the guideline when we have to go for the full validation, partial validation, and cross validation, which we will discuss in the next video. Validation parameters, here I have just highlighted some parameters, that is selectivity. In that experiment you can check the plasma or blood or urine is clean or not, which are going to use in the method validation. Accuracy and precision, this is the joint experiment that is called the P&A batch. And that experiment we are measuring the method accuracy and precision. Next is the linearity. Linearity is always required to know the concentration of unknown sample. Next one is the recovery. Recovery, you can compare the recovery with aqueous sample. So how much drug is recovered from aqueous and matrix samples. Next, by that you can compare the recovery, and suppose you can say that the aqueous sample giving the 100% recovery, while matrix samples are giving the 80% recovery. Next is the matrix effect. Matrix effects means you are dealing with the drug is soluble in the matrix, so somehow effect is showing the in aqueous and in matrix. So there are set of samples are defined in the guideline, By that you can say that this is a matrix effect. Due to that, results are low or high. Stability. There are sets of experiments like benchtop stability, batch size determination, long-term stability. Uh, Sets of experiments are defined to prove the stability, short-term stability, stock solution stability, working solution stability. So you can prove the stability by different experiments. Work plan. How we can plan our work? The first step is the literature review, as we have discussed. Next is the analytical technique selection. As I have already told you, the extraction technique, chromatography technique. Next is the development of method. So the development of method requires the some literature review. After the trial and error, you can develop the method. After that, you can select the validation of method. When you have tried a lot in the development phase, then and then you have to decide to go for the validation. After that, all the data I'll compile, and we can go for the result and conclude our drug. Thank you very much. This is the introduction for bioanalytical department, or you can say the bioanalytical method validation. If you like my video, you can like my channel and subscribe my channel. Your like and comments will inspire me to make more and more video. I will explain you in detail uh, each parameter, each experiment, and which samples are required to perform that experiment according to guidelines. So everyone will be the clear with bioanalytical method validation. Thank you very much.